Hello learners, welcome to our today's lesson, and in our today's lesson we are going to talk about food taste. And make sure if you've not subscribed to our channel, make sure you subscribe so that you might find such videos. So let's talk about the essentials of knowing a food taste. Number one, let's list all the types of food taste we know, whereby we have taste for starch as the first one. Number two, we have the taste for reducing sugars. We also have taste for non-reducing sugars. Number three, we have taste for protein. We also have taste for vitamin C. We also have taste for lipids. So the types of food taste we have are 6. Number one, starch, reducing sugars, non-reducing sugars, proteins, vitamin C, and lipids. So one thing you need to understand when trying to read on food taste, understand the reagents of testing each and every type of food taste. For example, when you are testing for starch, the main reagent is iodine solution, and the color of iodine solution is brown in color. That is number one. So each and every time you see the iodine solution in any type of food taste, make sure you each and every time you see iodine in each and every practical work, you will understand. If you see iodine in a question talking about food taste, just know they are referring to starch. If you see iodine in a food test, you are automatically required to test for starch. That is what you're supposed to know by now. Then in test for reducing sugars, we have the reagent known as Benedict solution. We have the Benedict solution. In testing for non-reducing sugars, we have number one, HCl, which is the hydrochloric acid. We have what we call the sodium hydrogen carbonate. We also have what we call the Benedict solution. So those are the reagents that you're required when you're testing for non-reducing sugars. And we also have tests for protein. And we have the reagents including sodium hydroxide. And we also have copper 2 sulfate as another reagent. In testing for vitamin C, we usually have the DCPIP. Another name, and the full abbreviation of DCPIP is dichlorophenol indophenol. That is the full abbreviation of the word DCPIP. And then in test for lipids, lipids are tested in 2 ways. Number one, the emulsion test. And the requirements include what we call ethanol, an organic solvent, and what we call water. That is usually distilled water, yes. And then another test is known as the grease spot test, whereby you require the filter paper, source of heat, and you also require to have what we call maybe the distilled water, whereby you can be able to do the controlled experiment. So basically, as a student, you should be able to be knowing each and every reagent that is used in each and every type of food test. And if we can start with the test for starch, let's talk about test for starch. Now, in test for starch, we've known the reagent is iodine solution. So the procedure, you should have the procedure. So the procedure of testing for starch is 1 to 2 ml. You can say to 2 ml of the food substance you might be given food substance X, food substance Q, food substance Y. So you are supposed to name it. Let's say Y. Yes, you're supposed to mention it. So to 2 ml of food substance Y, add 3 drops of iodine. And lastly, observe the color changes. So simply what you need to understand here is that I used 3 drops. I I wanted to be specific. So you might say 2 to 3 drops. Just be specific and say 3 drops of iodine and observe the color changes. So that is the procedure. It is constant throughout. The procedure will never change. So, and what you need to understand, if you write the wrong procedure, you get the wrong observation and you get the wrong conclusion. So we also have the what we call the observation. And there might be a negative observation and a positive observation, whereby if there is presence of starch, you'll see a blue, black color change. And again, if there is no blue, black color change, that is a negative observation. So if you observe a blue, black color change, if the substance has starch, so your conclusion will be starch present. That is all about the test for starch. Yes, you get the correct procedure, the correct observation, and the correct conclusion. So if you get the negative observation, automatically your conclusion will be starch is absent. And that is all about the test for starch. Now, let's talk about the test for reducing sugars. And in test for reducing sugars, the main reagent is Benedict solution, if you can remember. And this is the procedure for the testing for reducing sugars procedure. So number one, what you need to understand is that to 2 ml of the food substance, to 2 ml of the food substance, add an equal amount of Benedict's solution. Add an equal amount of Benedict's solution. After adding an equal amount of Benedict solution, heat to boil. So the mixture of the food substance and Benedict solution, you heat it to boil. So some people might heat, but don't leave it to boil. So when you heat it, the color will not change to the maximum. So you might heat, if the color was supposed to change from green to yellow to brown, it changes from green to yellow because you never gave that reagents time to heat together with the mixture of the food substance to boil. So make sure you heat to boil, and then after heating to boil, observe the color changes. Observe the color changes. Now, the observation you're supposed to see is the color either changes from green to yellow or to brown. And one thing you need to know is that if the color changes to green, it indicates little amount of reducing sugar. So the color might change to green when there is little amount of reducing sugars. Yes. So what you need to understand is that the color may change to green, which indicates little amount of reducing sugars. Then it might also change to yellow, indicating moderate amount of reducing sugars. And it might also change to brown, while this one indicates high amounts of reducing sugars. So these are basically the observation you're supposed to see. These are the observations that you're supposed to see if you're testing for the presence of reducing sugars. And of course, the conclusion is so easy. If you see those color changes, you automatically say that the reducing sugars present. Reducing sugars present. That is all about that. Now, let's talk about the test for non-reducing sugars. And this one is almost similar to the test for reducing sugars, but there is a change of few reagents. But almost the reagent also Benedict solution is used in this procedure. So the first reagent we use in test for non-reducing sugars is known as the hydrochloric acid. And now the procedure is to 2 ml of the food substance, to 2 ml of the food substance, X, Y, or Z, add 3 drops of HCl. So the next thing you do is heat the mixture and let it cool. Let it cool. Heat the mixture and let it cool. After the mixture has cooled down, add sodium hydrogen carbonate. Add sodium hydrogen carbonate. Dropwise until fizzling stops. Then after doing so, now add 2 ml of Benedict solution and observe the color changes. Yes. So after you've added the 2 ml of Benedict solution, just observe the color changes. And now the color will change either from I said it's almost similar. So the work of HCl is to hydrolyze the non-reducing sugars to what we call the reducing sugars. That is why we use it. So that after we have hydrolyzed the non-reducing sugars to reducing sugars, we can now test for reducing sugars by the use of the Benedict solution. So simply, what you need to remember is that it is like we're testing for reducing sugars. But what do we have to do? We must therefore hydrolyze these non-reducing sugars to reducing sugars so that the testing should be easy. And the conclusion, and I mean the observation, if there is presence of reducing sugars, of course, there will be a color change from green, yellow, or brown. And these, or the green indicates little amount of reducing sugars, yellow indicates the moderate amounts, brown indicates high amounts. And that is all about that. So this one, the conclusion should be there is presence of reducing sugars. There is presence of reducing sugars because we have hydrolyzed the non-reducing sugars to reducing sugars. And that is all about that.